I've never had it notify like that before. That's awesome.
Really?
What are you recording?
It might be a setting with my Zoom, a Texas State.
Yeah, I like it.
Yeah, I can actually turn it off, but then it's kind of creepy if I start recording and don't let other people know. Yeah, we're going to record the presentation, Nico, that Audrey Brumback is going to do.
I'm just going to close all my embarrassing windows so that when I share my screen, it's all kosher.
Yeah.
Should I? Yeah, just let me know when I should start. I'm happy to,
Okay.
Whenever.
Sandra, were you going to introduce, or do you want me to?
You can go ahead.
Okay, we'll get started and I'll introduce Dr. Audrey Brumback. She's an assistant professor in the Department of Neurology and Department of Pediatrics. She's a board certified pediatric neurologist at UT Health Austin Pediatric Neurosciences, and sees a lot of kiddos coming through for autism and other neurodevelopmental or issues or neurological issues. So, but she has some really interesting research, and that's what she's going to talk to us about, developing novel therapies and for brain dysfunction. But I'm not sure exactly what Audrey will be talking about today, but she will share that with us. So I'll turn it over to you.
Yeah, thanks, Sandy. Yeah, so I'm actually not going to talk about any of my research today. I wanted to use this to educate people about sort of as a clinician, how do we think about genetic testing in our patients? And so let me just get rid of that screen. Okay, and I'm assuming everybody can see the slides. So yeah, so we'll talk today, I'm going to talk about autism, but really this, the conversation really applies to kids with, a wide array of neurodevelopmental challenges. And so we can talk at the end if there's specific questions about, about particular different, clinical presentations that people are interested in knowing kind of the approach. So I'd like to start today by talking about just noting the diagnostic discrepancies that are happening in autism. We'll talk about what causes most cases of autism. Hint, hint, it's genetics. And then I'll explain sort of, how the genome works and how we go about detecting genetic changes and what the benefits and risks are of doing that genetic testing. So, if you're not a little white boy, you're much less likely to be diagnosed with autism. So this is just one study comparing white kids and Black kids and when they were diagnosed with autism by specialists. So compared to white kids, Black kids were 2.6 times less likely to get the correct autism diagnosis on their first visit. They were 5 times more likely to get an adjustment disorder diagnosis and 2.5 times more likely to receive a conduct disorder diagnosis. And so, we're swimming in racism, and this is just one component of that, and how it impacts how we perceive people's behavior. And so I just want to, for the clinicians in the room, if you have somebody come to you with an adjustment disorder diagnosis or a conduct disorder diagnosis who's not white, I want you to think twice about whether you think that's the correct diagnosis, and just think twice about, could this be autism? Could this be explained by another diagnosis? In families who have low educational attainment, so where the family, the parents, have not graduated from high school versus parents who have higher education degrees. There's a real spread in the rates of diagnosis for autism as well. So here in this graph, we're showing the prevalence of autism as a function of time. And then in the colors the population's been separated into 3 tiers of educational attainment, with green being the highest and red being the lowest. As you can see, just for a period of 8 years, for every at every time point, there is a lower diagnostic rate in parents who have a low educational level. So again, I think, when we're to sort of overcome this tendency, when you're seeing a child whose parents, are not highly educated, think about autism, think about, whether something has been written off as not autism that might actually qualify for autism. And I acknowledge that this discrepancy, all of these discrepancies are not just at the level of the individual clinician making decisions, but at a more, structural level in terms of access to care, et cetera. But I do find just in my own clinical experience that the kids who are Black and brown, the kids whose families are not well educated and know how to advocate for their children, and girls are not receiving autism diagnoses when they should be. So, right, so, the same goes for females. So boys are 4 times more commonly diagnosed with autism than girls. And surely there are probably some genetic reasons for that. There's biological reasons for why boys may have a higher rate of autism than girls. However, I have been seeing a lot of girls lately who, clearly have autism and it was just never recognized. So I teach all of the residents who come through my clinic that you can have 2 children with the exact same symptoms walk into the clinic room. The boy walks out with a diagnosis of autism. The girl who has the same symptoms walks out with dual diagnoses of anxiety and ADHD. And so I teach people that if you see a girl and she has dual diagnoses of ADHD and anxiety, to think about whether that could be autism. I'm seeing lots of teenagers and adults who, have been basically misdiagnosed their whole life. And I think that we need to have a fresh look at how we're approaching autism in girls. Okay, so now that we've discussed all of that, and I can step off of my soapbox, we can talk about what causes autism. And so, this is, I sort of pose this as a very simple question: what causes autism? With the understanding that that is I don't exactly have the answer to that question, but I can educate you guys about, what's known and what our current thinking is based on research. It's not vaccines. Okay, so the upshot is that, as far as we're aware, genetic changes are really responsible for most cases of autism. And so, so what is that based on? So in the general population, let's say, that there's about a 1% risk. And that, I know that this has been changing, and, but I think that 1%, most people can agree that that's sort of a conservative estimate of the risk of autism in the population. If your identical twin has autism, your risk goes up to 90%. So that's for people who have a family member, you have 2 kids, they're both affected. That's actually not the most common scenario. The most common scenario is where you don't have a family member who's affected. And so when we look at families where in blue you have unaffected family members, and then in orange you have an affected family member, what we're seeing is that the genetic changes that we're seeing in the affected person are arising spontaneously. So they are new, they're de novo in that affected person. So what does that mean? So genetic does not necessarily mean inherited. There are a lot of genetic changes that take place at the time that sperm meets egg, and you go from a single, the 2 half cells into a full cell. That full cell divides into 2, and then into 4, and 8, and 16, and on until you get, a fetus. And during that early developmental process, there's a lot of changes that take place in the genes. And so at that stage, at that very early developmental stage, you can have changes that happen in the genetic code that have nothing to do with the parents, essentially. And so when we test the children, and then we test the parents, we find that these genetic changes are not present in anybody else in the family. And so we conclude that these were de novo genetic changes in the affected person. So let's talk about what, if we're going to identify these changes in the genetic code, how can we conceptualize that? So I think of our genome like a library of books. And so in that library, you have bookcases, those have bookshelves. On those shelves are books. Within those books are chapters. Within those chapters are paragraphs. The paragraphs are made of sentences. The sentences are made of words, and the words are made of letters. And so we have, the entire genome represents the bookcases, and we have the ability to analyze the contents of those bookcases, the contents of that library at these multiple different levels. So to start with the sort of the most elementary change that can take place. So if we have a sentence, the cat meows, we can have a misspelling in one of the letters of those words. And so we go from the cat meows to the meows. And you can see that that has changed the meaning of that sentence, and it no longer makes sense. We can also have deletions where an entire word has been deleted. So instead of the cat meows, it's the meows. And then finally, we can have changes where we duplicate, we copy and paste a word, where we have the cat meows. So these are the different types of changes that we'll talk about. And this essentially covers the different types of changes that we test for. We look for misspellings, we look for deletions, and we look for duplications. And so I'll tell you about the names of the tests that that assess for each of these types of changes, and what are the types of genes that we identify as being changed by these different mechanisms. So, so we have sort of the 3 levels of testing. One is karyotyping, the next is called chromosomal microarray, and the third is called exome sequencing. And so I'll walk you through each of these types of testing because they ask different questions. So the first, so karyotyping. Karyotyping detects duplications or deletions of big pieces of DNA. So if we think of our library, analogy. The karyotyping is really picking up changes in entire bookcases or in entire bookshelves. So we can have a deletion of a huge chunk of DNA, or we can have a duplication where a large piece of the chromosome has been copied and pasted. So this is karyotyping, and this used to be really the only type of testing that we had, up until maybe 20 years ago, where we were able to look at the chromosomes macroscopically and see if there were big chunks missing, or if there's an extra chromosome. So the technique that we use today is called chromosomal microarray. And so the idea here is that we're able to also detect duplications and deletions, but now we're able to do it in both the big pieces of DNA, so the bookshelves and the bookcases, but also at the level of, books, chapters, paragraphs, sentences, and even words. So we're really able to take that concept of detecting these types of changes and do it at a much finer scale. And so for a small deletion, we might expect to see the cat meows turn into the meows. And for a duplication, that same example I showed you before of the cat meows. A larger version of that might be, the erasure of a few lines of text. A duplication would be, an entire paragraph that's been copied and pasted. So this is what we are able to detect with chromosomal microarray. And this is really the first step in our genetic testing. And this is for both kids with autism and also for global developmental delay and intellectual disability. So what I described to parents is, how do we actually do this, is that we take the genetic material and we chop it into little pieces. And then we ask, are any of those little pieces deleted or duplicated? And so one example of a gene that is genetic changes that are often detected using this technique is with the 16p11.2 region of the chromosome. So just as another sort of educational aside, we talk about chromosomes, we talk about them as the number, and then P or Q. P is the short arm, Q is the long arm. So all chromosomes are sort of asymmetric. And then the other numbers are describing where along the arm of the chromosome the location is that you're talking about. So interestingly, with this particular location in the genome, duplications of this region lead to macrocephaly, hypotonia, and hyporeflexia, whereas deletions lead to microcephaly and hyperreflexia. So it's really fascinating that we have this kind of dose-dependent influence of the genes in this region. If you have a duplication, there's too much of the genes in this region. You have one end of the spectrum where you have a large head and decreased reflexes, decreased tone. If you delete it, you get a small head and increased reflexes. And so the next step after we've identified the region is to ask, well, what are the genes that live in that region? And so for this region, there's at least 3 really important genes that we know have to do with brain function that I've listed here. So how often do we find a specific etiology when we do our genetic testing. So actually, I think I got my slides a little bit out of order. Let's see here. Yeah, so let me go here. So before I talk about the how successful we are. So we've talked about karyotyping, where we're looking at big chunks of DNA, microarray, where we're able to look at both big chunks and small chunks. And now we're going to talk about exome sequencing, which is the next step in our diagnostic pathway. So this approach asks a really different question. So we're asking if there are any misspellings in any of the genes. So we're looking at the level of letters, and we're only doing it in a select part of the genome. We're doing it in the part of the genome that's called the exome. So you might have heard the term junk DNA. That was sort of a popular term back, maybe in the early 2000s, where it was really referring to most of our genetic material. So most of our genetic material, like 99% of it, does not get turned into protein. And that's really, the going from gene to mRNA to protein is really the fundamental way that we understand that genes do their business, is by encoding the instructions to make proteins. And then those proteins are the enzymes and all of the structural components to our body, essentially. And then that's only 1% of it. The rest of it, we used to call junk DNA. Now we understand that that's not junk. It's just that we didn't understand what it did. And so we gave it this pejorative term. But because this detecting misspellings in genes and in genetic material is so labor intensive, we only do it on this 1% of the genome, the part that's sort of the business end of the genome that gets turned into protein. We are moving into a phase where we're going to be able to do this for the entire genome, the other 99%. But that's sort of not kind of prime time right now, but exome sequencing certainly is. So, okay, so now I'm going to go back and talk about how often we find a genetic etiology. So with microarray, we will detect changes about 15% of the time. When we add on the exome sequencing, we're able to get a diagnosis about 30% of the time. And then the genome sequencing, which is really mostly done on a research basis right now, but is starting to be available clinically, we can get an additional 1 or 2%. And I imagine that as our understanding of that junk DNA, as our understanding of that, the rest of the genome improves, we will be able to detect changes that we think are significant for brain development. So what are the clinical features that make us more likely to find a genetic cause? Being female, having a low IQ, having seizures or epilepsy, having a large or a small head, having syndromic features where there are other, say, organs involved, and then remarkably delayed walking. So, I've just gotten done telling you about genetics. I do want to acknowledge that that there are other ways to influence the genome besides having misspellings, deletions, or duplications. And one way is by adjusting which genes are basically available to be for access. So what I'm talking about is broadly termed epigenetic changes. So our chromosomes are packaged in a way that they are nice and compact. And in order for the enzymes that need to read the genes to access those genes, the genetic material has to be kind of twisted and stretched and sort of exposed so that those enzymes can do their thing. And so, as you might imagine, that whole process of how the genetic material is packaged, you could really influence things that way if you changed which genes were kind of available or not available, even regardless of whether those genes are like the correct versions of those genes. Even just by saying, you know what, we're not gonna let this gene be accessed, that would be sort of kind of equivalent to just deleting the gene. If you can't access it to, transcribe and translate that gene into a protein, then it's almost as good as not having the gene at all. And so there are medications that influence this process of what genes are accessible. And one of those drugs is a commonly used anti-seizure medicine called valproic acid. And so we, I think, have very good evidence at this point that when a fetus is exposed to valproic acid, there is a higher chance that that person will go on to have autism. And so this is a study looking at all children born during a 10-year period in Denmark. And basically the risk of having autism based on what anti-seizure medicine you were exposed to in utero. And the increased risk occurs when you see the boxes and the lines not overlapping that center dotted line. So you can see that valproic acid is the only one out of these 5 where that what we call the hazard ratio, that increased risk that you have doesn't overlap that center line. And so this and other studies, I think, give really nice evidence that having one of these exposures that can change how your DNA can be accessed, which is what this drug does, can influence brain development and lead to neurodevelopmental challenges. So interestingly, valproate was banned in Europe based on these studies. And that's a topic for another discussion, but for another time. But based on this, in Europe, valproic acid is forbidden to be used during pregnancy, except when no other treatment is available. So yeah, so I think that this is a very interesting sort of modifiable risk factor that clearly people in Europe are taking very seriously. And it's also something that we in the US take seriously when we're working with people of childbearing age. Okay, so we do this testing and we identify a genetic change. So then what do we do? So the first question we ask is, was this something that was inherited, or is this something that is brand new in the child? So we're really asking, do the parents have the same genetic difference? If the answer is yes, and the parents are unaffected, then we say, you know what? It's probably not the root cause, because if this genetic change is that bad, then why is it that the parents are fine and the child isn't? And that's, sort of over exaggerating the thought process, but it is that sort of idea. And then we say, okay, well, if this is a new genetic change, then we say, well, is this in a part of the genome that's important for brain development or brain function? You can imagine that a person could have genetic changes that are maybe important for, toenail growth that aren't going to influence brain function. And so, so we really, look at the function of the gene and say, does this, do we think that this actually is related to the reason we did the testing? So what are the risks of doing this? So the first is cost. Depending on a person's insurance, the cost of this can run from $0 up to, thousands. Medicaid now pays for that first line testing, the chromosomal microarray. The exome is really only available to people who have commercial insurance. And even then, it can cost thousands of dollars, depending on if you've met your deductible for the year. And then there's the idea that the risk that we will find answers that we don't understand yet. We don't understand what all the genes do. And so we may find an answer that we don't quite know how to interpret. And then I think the third major risk is finding something that we weren't looking for. So when we do the exome sequencing or the chromosomal microarray, we're looking at, we're not just looking at the genes that we know are important for brain development. We're looking at genes for everything. And so, we might pick up a difference in a breast cancer risk gene. And so before we do this testing, we always ask the family, if we find something like that, do you want to know? Because some families would prefer not to know, and some families want to know everything. And so finally, I think I just want to sort of, when I counsel families about the risks, I really do spend a lot of time talking about this idea of the variance of unknown significance. So, you have sort of your green zone where you have something that's not causing a disease, you have like no changes, or you have the red zone where you find a change in a gene, it's a new change, and it's, we know it's in a gene that's important for brain development. But then there's a lot in the middle, in that yellow zone, where we don't understand the significance. And I just tell the families, this is where, when you come back to see me, every time I will look through the scientific literature and update myself on what the new, information is regarding that gene. And I can, educate you about whether we have any new information that's going to help us know whether this is significant or not. Okay, so why do genetic testing? So I think that there's several reasons. First is anticipating and managing other medical issues that, that we know are associated with specific genetic changes. The second is, sort of along those lines, is improving the understanding of treatment and prognosis. As we understand the natural history of people who have different genetic differences, that can help us tell the family, about what the future might hold. There's a lot of guilt, I think, associated with, parenting in general, but the idea that a parent did something wrong or didn't do something that they should have done, I think really is a big challenge for a lot of families. And, there can be blame put on the mother for, having taken a Tylenol pill, in the second trimester, things that I think that what I've found is that having a genetic diagnosis can often relieve a lot of that anxiety about, whether they had done something wrong. To sort of have an alternative explanation to that, I think is really relieving of that guilt. As we understand more about the genetics of our neurodevelopmental disorders, foundations are created to support research and support families, like Haley with our SETBP1 Society, to really, engage families to share resources and knowledge. When we have a patient who we don't understand why they are developing differently, as neurologists, we really want to find the answer. We want to understand the biology. And so if we do genetic testing and have an answer, then that can prevent a lot of further testing that we might do if we didn't have the answer. Counseling regarding recurrence risk is also important. And then sort of along with that, preventing recurrence through screening for carriers and prenatal testing, especially for autosomal recessive disorders. So we typically do the chromosomal microarray, and then if that doesn't give us the answer, we do the exome sequencing. You see this child here having a Q-tip placed in his mouth. This is the way that we collect the genetic sample. It's just through a cheek swab that is really no more difficult than brushing a child's teeth. We collect these routinely in our clinic, and it's rare that we have a child who doesn't let us collect the sample. So what kinds of genes are involved that we find related to neurodevelopmental disorders? So they fall into different categories, but the main sort of themes are that we see changes in genes that regulate other genes. We see changes in genes that regulate how neurons communicate with each other. We see this in both the major types of neurons, both the go neurons and the stop neurons, so the excitatory and the inhibitory neurons. And then we see this in newborn neurons, as well as genes that are expressed in neurons as they are maturing. And so it's not just which gene, it is where and how that gene is different. So if we focus in on one of these genes, SCN2A, we see that where the genetic change is within the gene makes a difference as to what the clinical outcome is. So here I'm showing you, basically, if you took this gene, which encodes a sodium channel, which lives in the membrane of a neuron, and you sort of, it's sort of a long string, and it folds on itself and creates pores in the membrane to let sodium in and out of the cell. And so in this view, we sort of have flattened it. So we are looking at the full string and kind of where it meanders in and out of the membrane. And you see these blue and red dots. So these are all locations in that gene where people with neurodevelopmental disorders have known mutations. So all these dots represent places in that gene where there are patients out there, there are people out there where we have found that person has a specific genetic change. And each person essentially is represented by one dot. And what's, I think the point that I want to make here is that depending on where in the gene that dot is, that dictates in large part what type of symptoms you will have. So for children who have epilepsy, we see changes in where the red dots are. Kids who have autism or intellectual disability have the changes where the blue dots are. And these are, so you have a genetic change in the same gene, but what that genetic change actually is on like a detailed level really makes a difference in what type of symptoms it will lead to. And similarly, we can have genetic changes in genes that don't actually impact the function of that protein. So, you know, so that's why when we get an answer, we see, okay, we have a change in a gene. That gene, genetic change is not present in the parents. So we think that it's probably, you know, a new genetic change. It's in a gene that has something to do with brain function. Well, is it then in a part of the gene that's actually going to cause that gene to not work? Okay, so I'd like to end there and open it up for questions. So today we've talked about diagnostic discrepancies between girls and boys, Black and brown versus white kids, and families with high educational attainment versus low educational attainment. We talked about genetics. We've talked about epigenetics, which is just the access to the genes. We've talked about how we detect those genetic changes by looking for duplications or deletions or misspellings. And we've talked about how many of those changes are in genes that are important for brain function. We've talked about the benefits and risks of genetic testing and as part of that conversation that we have with families when we offer this. So one more slide. This Spark for Autism is a free genetic research study for people with autism. You can give the family the website. They go onto the website and their portal, create a profile, and then the organization sends the family a kit to do a cheek swab at home to collect a saliva sample. And then the family is enrolled in the study. And what I like about this is that you also get plugged into the newsletters about you know, the latest in genetic research, and it's for the idea is that they're trying to collect 50,000 families, and they're halfway to their goal at this point. So this is a really reputable organization, the Simons Foundation. And so I encourage all my families to at least go to the website and look into it. Okay, so that's everything. I'd like to stop sharing and take questions.
Yeah, there's a couple of questions in the chat that you could, since we're a group, you can go ahead, you guys can go ahead and ask your question. Like I think Lauren has a couple. You want to go ahead and ask your question, Lauren?
Yeah, sure. I have another meeting. I'm gonna have to jump off in just a second, but I was curious about the reporting of the genetic results. So if you know all the things, like they have an increased risk for breast cancer, is that something that will stay in their file? So when the child becomes an adult, could they access that information if they wanted to know, or would they have to do more genetic testing? And then if it does stay in the file, does that impact the child's ability to get life insurance? I don't know if you know that, but there's just a lot of questions about.
I'm embarrassed that I don't know the answer to these questions.
Okay.
My understanding is that that information goes nowhere. It stays with the organization, the company that does the testing. But I don't know what happens when the child turns 18. It's such a good question, and I do not know the answer to it, and I will find out.
It's okay. I recently had a bunch of genetic testing done on myself. And so I'm like very curious about how this all, like where the file lives and where the data live and how people can access it and all that. So, and then the other question was, is this ever part of routine monitoring for siblings? So, you know, when families are concerned about routine or when they're concerned about their younger siblings, when they have an older child diagnosed, is this something that you would offer? Is that something that insurance would ever pay for? That we could find out early if the sibling was at risk?
Yeah, so siblings, you know, I sort of told you the extreme example of how you have an identical twin, and that chance of also having autism, you know, is raised to 90%. But even in non-identical twins, in siblings, the chance of having autism is increased. And so what is our approach to that? So I have not tested siblings when the genetic change isn't gonna change immediate management. If it did, then I would. So for instance, with disorders like tuberous sclerosis, that ends up being mostly a sporadic disorder. But there are, you know, medical issues that happen early on that, you know, we would want to know about, that we could start treating. And so if it's something like that, where you would be able to help the person, you know, starting early, and that would be beneficial, then yeah, we would offer that testing. If it's something where that's not the case, then I don't, and this is where we collaborate with the genetic counselors, because they can delve into this with the family and understand, you know, sort of the current insurance structures and what is covered and what's not. So yeah, I would say that this question ends up getting addressed mostly by the genetics counselors. Yeah.
Okay, thank you.
Sherry, do you have other comments on that?
Hi, excellent presentation. That was so good. Thank you.
Oh, thanks.
That's a great question about where all this stuff lives in the chart and who has access to it later. I do believe that the patients will have access to it after they turn 18. What I sometimes worry about is the patient's family members who would have access to it, like a brother or a sister, or how do we reference like a brother or sister has this genetic difference, and therefore maybe we're doing testing in this patient without actually mentioning that sibling, because you are kind of then sharing someone's medical information with someone who's not them. So I always get a little bit spooked at that, but I've had a lot of people try to get life insurance before they get any genetic testing, because what I have been told that whereas you cannot have your genetic testing be held against you as a pre-existing condition for medical coverage, it can in certain circumstances, I guess, be factored in when applying for life insurance. So I had a family just get life insurance on their like 5-year-old. You know, it's just starting early, I guess, but before we did genetic testing. So I thought that was pretty interesting. So, yeah.
Yeah, that's why I asked, because that was the recommendation my genetic counselor gave me, was before we do this, you should make sure you have all your life insurance finalized, just in case, right?
Yeah.
Yeah, so it does have a big impact. I think it's important for families to know that.
Yeah. We're trying to get a genetics counselor for our clinic. We have an ad posted because right now, in order for us to access genetic counseling, we have to refer to genetics, and their wait list is, you know, eons long. And so I think it'll really help us to provide better care once we have our own genetic counselor.
Do you ever use My Gene Team?
What's that?
Do you ever use My Gene Team? They are, so it's interesting. I guess whichever genetics lab you go through has to have a secondary, I guess, unaffiliated genetics counselor availability. And so, yeah, My Gene Team, I don't want to promote them. I'm sure there's tons of other genetics counselor groups out there that anyways, like, for example, we get our reports back and they say, if you would like genetic counseling, you can contact this group. They're not affiliated with the genetics lab in any way, shape, or form. They don't get a kickback from referrals. But in order for, I guess they passed a law at one point in time, in order for these tests to be run without a geneticist actually ordering them, the lab just had to tell the consumer that there was access to a genetics counselor if they wanted it. So this is how we came into contact with My Gene Team. But they do have to have a referral from a physician, and then they can set up and they can talk about the genetic, findings. I don't know how good they are or how good this tends to be, but that's what we've been using. Well, because it is hard to get in with genetics.
One thing that I wanted to share is that, so I'm the new director of Texas Center for Disability Studies now, and they have a lot of existing programs that I didn't create, that are going on. And one of the programs is they get funding from DHHS in Texas to provide genetic counseling. I'm not counseling, but information to different communities throughout Texas. And so they're working with Baylor. Actually, they contract with Baylor, one of the genetic docs there. And then there's a parent who works for TCDS that has a child with a genetic disorder who is on that presentation team. And they go around and do presentations. And I don't know if it's similar to what you present or not. I haven't seen their presentations, but and they have recordings of them. So I'll have to share it with this group so you can kind of see what they're doing. But then also, I mean, if we have ideas of underserved communities that really need this information, we can kind of steer them in those directions. Because as Audrey mentioned, there are people who can access certain things and others that can't. But even information is pretty hard to access sometimes.
Yeah, and that's one of my questions I was going to have, or questions I had, was whether there are certain, families that are resistant or who outright do not agree with following through with genetic testing, or are most families usually on board with that? Or kind of, because I know in Chicago, when we were doing, we would refer most families who would evaluate for genetic testing, but sometimes that, they didn't have insurance, or so then that would be the issue. But I don't know here if, there's some of that resistance with families.
Yeah, I actually have had very little resistance from families. I feel like the vast majority of families that I offer this to are like, yeah, whatever. I mean, sort of almost like maybe they shouldn't be so, default to just do what the doctor says. Which is why I really try to go through the risks and say, but I'm clearly from Lauren's question, I'm clearly not counseling them on all the risks, and so, yeah, I think, I don't know, Sherry, what's your experience? I find most families do it.
Oh man, I'm so sorry. I'm listening to two things. I'm always concerned that I under counsel them. I'm always concerned that I'm not telling them enough. We definitely talk about the VUSs and what could potentially happen there. But it's so interesting that you kind of do that part about what if we get information that you don't want to know. And I feel like sometimes we don't always go over that to, in depth. and I just had a really interesting situation this morning where we went ahead and tested the child. They had a variation of uncertain significance. And then we tested the parents, and then it came out that it was contributory from the father, but it was something that would probably affect potentially fertility. And it already, so it was so interesting. And so it's like, well, who do I give this information to? Do I give it to dad, or do I give it to mom? Or, you know, like, obviously I give it to dad, but do I give it to mom too? You know, so it just opens up so many, you know, different cans of worms. Because I mean, I'm not a geneticist, so it's kind of hard to know. But I love that we have these first line tests though, because like Audrey said, wouldn't it be great to kind of know with this test and then not go down that diagnostic pathway where we take all sorts of blood and urine and other samples. It's just very, very helpful in the beginning. And I think sometimes parents do push back because, and their biggest issue is cost. But we have just started doing the estimates for how much we're going to owe out of pocket before we even do the swab. And I'm a little bit on the fence about whether or not that's good for the family, because I feel like some of them want to know and some of them feel like it's just too much information right then and there. And then the other is just like, well, we don't know, it's not going to do anything necessarily. You can't change his diagnosis. It is what it is. And so they push back against it from that point of view.
Yeah.
I think in Chicago, I know we had a couple of cases where, and this may have been with the pediatrician that I was working with, where the mother was resistant because it would also open up issues with paternity as well. That sometimes, you know, it's not something that you would want to discuss with everybody in the room. So it's definitely, you know, something else to consider.
Yep. Haley, I'm curious to know your thoughts.
Yeah. So I have for those that don't know, I'm Haley Euler. Dr. Brumback mentioned SEPBP1 Society, and I run that nonprofit. And so I have a son with special needs who has a monogenetic neurodevelopmental disorder. So we did go through a geneticist, and I did feel, we talked to the genetic counselor first, and these things you're bringing up, it's been a while. Let's see, my son's been diagnosed just over 5 years ago, but they did bring all these things up. And so about paternity, about USs, about there could be results, do you want to know or not know? It was I think the genetic counselor did a good job of going through all of those things. And then I'm of the mind of, regardless of if there's a treatment, you know, I think Dr. Brumback, you went through a lot of valid reasons of why you would want to go through genetic testing, which a lot of them don't cover whether or not there's a current treatment available, right? And to say it doesn't change treatment isn't necessarily totally true, because you don't know what all those other extenuating circumstances are. You don't know what additional communities they will now connect with, what they will learn from those communities, what other traits they're going to, they might share with others and have other sort of ways to address those differences. So I personally am of the mind of, like, I promote, of getting genetic testing, but definitely going over all the concerns, because you can learn about other things. Maybe you learn that your child has like an organ difference you didn't know about. So now you need to go and test for it, which can add some additional fear, but however, it also is giving you more knowledge on knowing how to help your child. And I'm speaking mainly from the child perspective, because that's mainly what we're looking at. Most of our kids are diagnosed as kids. So I was going to say one thing of hearing, you both talk. I think the role of a genetic counselor, like Dr. Brumback, I'm very excited you're getting a genetic counselor. I was actually, before you started talking, I was looking that up. So as you were talking previously, I was looking up, is there a genetic counselor linked with you? And, I was checking on that, and I'm like, I don't see anybody linked and connected. And so I was trying to figure that out, because I think that's where a genetic counselor really comes in and helps to explain all that stuff. And then when the geneticist presented it, then in our specific scenario, the geneticist presented, you know, what all limited information she knew at the time. But I think a genetic counselor is a very important role, and VUSs, they're being brought up in our community. They're a hard one. There's no clear answers. And then Dr. Brumback, we're also seeing some mosaicism, so it's not black and white. I'm like, some of these things are, but in general, so we're having, my son's condition's very rare. It is linked, some of our kids have autism, some have IDD. But in a lot of the rare groups, we're seeing patients with VUSs and then sometimes inherited, and it's this big fuzzy space of, is it really that gene or not? And it seems to be, I think, with the advancement of testing, these are becoming more prevalent without a lot of knowledge around it to really guide. So we have numerous community members who are sitting in a fuzzy space. And so we just recommend they stay connected with the geneticist. Dr. Brumback mentioned Simon's Foundation. We're connected through Simon's Searchlight, which is one of their initiatives. So we recommend they stay connected with them because they will do periodic reruns of their results to see maybe the next year, there will be more data to know, is it going to become more likely benign or more likely pathogenic? But did you have any other, I clearly had several things to say about that, but was there anything more specific that I didn't answer that you're wondering about?
This is exactly what I was hoping to hear.
Okay.
I have a question. How often are VUSs kind of reinterrogated? And is that something that the family has to initiate? And then are they just contacted through whatever, you know, data they have? Do they have to actually go in and seek those answers? How does that process work?
Yeah, in my experience, I initiate that as a clinician, and the sort of recommended frequency of doing that is about every 5 years to do those reinterrogations, and that will not only sort of maybe change that VUS versus, you know, whether it's pathogenic or not pathogenic component for changes that have already been identified, but it can also identify changes that they might have seen but didn't think, you know, based on what we understood about that gene at the time, it was relevant, and so it wasn't reported. You know, so it's not just what the result is that you already have, the significance of that, you also get additional information. I think one other component to this is I have a handful of kids who have multiple different genetic changes that have been reported as VUSs. And there is the whole thing about like, okay, maybe having that in isolation isn't enough to give you a neurodevelopmental disorder, but what if you've got, you know, that plus this other one plus this other one? Would that combination be enough? And that is like, nobody knows. Nobody knows that. And it's, I mean, I think that's going to be a really important area of research, because like you're saying, you know, as we do more of this testing, we're getting more of these not interpretable results. I think that is oh, sorry, go ahead.
Oh, you're gonna say, I was gonna add one that we had a family member, a family join our group pretty early on, and her child had 2 VUSs, and then with about 2 years later, they rechecked the results. I will say that it sounded like it was parent initiated. I had honestly thought it would be more of an automatic process, and than it was. Like the way that it's been conveyed, I don't think that's automatically done at any places unless the parents request or the specialist is on it. But the results were reran, and that her daughter was identified with a different monogenetic difference that they determined was the issue. So she had 2 VUSs and 2 genes. They said, actually, it's not that. now, and those were both inherited from the mom. They found another de novo that they didn't even, call out initially. So I think that's an interesting one that I keep in mind for some of our families where they don't really know. I'm like, that may be the thing that happens in the future for them.
One other thing that I think is worth mentioning is that from a clinician's point of view, we want to do something to help, like we want to be able to do something. And I think that, this is one of the things that we can do. And so we end up doing it. Whether that's like right or wrong, but I do think that that plays a role, is that desire to help and that desire to have something to offer. And so I imagine that that does, influence the rate at which kids are getting these tests.
Excuse me, I have a question.
Yes.
So how exactly is this testing done?
How is it done sort of physically?
Yeah.
So it used to be that we had to take a blood sample. More recently, it's become very common for us to be able to sample saliva and sample cells that we can get from a Q-tip in the cheek.
Okay.
And so it doesn't require, I think that's another component to this that's made the testing much more common, is that it doesn't require something that's, painful for the child. And so it's sort of a, sure, why not? That activation energy barrier has been lowered so that the parents are like, yeah, why not? Just swab my kid. Yeah.
So if I have kids, if I have kids, and how will I know? Or I have autism. So if I marry and have kids, do you think they're likely to get to be passed on the autism?
Well, I think that things like your hair color and your eye color and, your sparkling personality, like all of these things, are partially genetic. And so I think that, much of who you are ends up getting passed on to your children genetically, but much of it doesn't. And so I would say at the time that you're thinking about, having kids, I would talk with a genetic counselor and find out kind of what the current understanding is of, how much of autism is transmitted, from parent to child.
Okay.
Because I think that it depends on, if you have a genetic change that was new in you, what's the likelihood that that'll be passed on to your child? And that's pretty complex. And so I would want to talk to a genetic counselor.
Are there any, oh, sorry, Nico. Did you have any other question before I ask my question?
No.
So I just had a question. Is there any genetic counseling program in Central Texas? We don't have one at UT, right? Genetic counseling.
Like a training program or?
Yeah, like a degree or.
I don't think so. It would be interesting to know, related to like Baylor and, the other medical schools, what they offer.
Yeah, maybe they have something.
Yeah.
The other one is in Houston, so it's not even local one, but where there's a affiliation. So.
Okay. Mm-hmm. Because that's something we might want to, bring in any genetic counseling students into the LEND program at some point.
Yeah, I love that idea. Yeah. Yeah, and I mean, now that everything's on Zoom, I don't know, the benefit of having access to a genetic counselor sooner is more beneficial than being able to wait and then do it in person. But I imagine that there are a lot of families that would be happy to do it over Zoom if it meant that they could be seen sooner. Yeah.
Yes.
Well, I hope it's such a, like, there's sort of the very simple ways to talk about these things, but it's so much more complex than that. And so, you know, the sort of way that I've described things today is really, you know, how I describe things to parents. And I know that that leaves out a lot of the nuance and, you know, that that I hope when we get our genetic counselor to learn, you know, how to communicate these concepts clearly, you know, so that I'm not failing to counsel my patients about important components of this. Because I do not counsel anybody about paternity. I've never done that. And it's, you know, I haven't had that come up clinically yet. And so I think that that's, you know, and then the other thing is consanguinity, parents who are related by blood. That is something that, you know, is detected as well. Yeah.
But you do such an amazing job breaking this down for those of us that are not, you know, have no background in this at all. So really appreciate that.
Thank you.
I did a version of this talk for the Project Skills for their webinar series.
Excellent.
So hopefully that'll be available for families. All right, well, we're well past the hour, so we should probably let people get on with their lives. But yeah, thanks. I mean, I feel like I've learned so much just from the questions you guys have asked, and, you know, Haley, hearing your insight, it's just always worth its weight in gold. So thank you.
Yeah, thank you, everyone.
Okay, have a good day.
You too.
Thank you, Nico.
Yeah, thanks, everyone.
Thanks, Dr. Brumback.